guys, I'm here to talk about, it won't let me do that. Oh, I need to share my screen. I'm sorry. I'm a little slow this morning. I'm going to share my screen. There it goes. Okay. Okay. So we're here to talk about the subcellular components and compartmentalization specifically. So when we're talking about cells, you've got to remember basic structural functional units of every organism. So every single living thing is made of one or more cells. All cells must have a plasma membrane. Sometimes we refer to it as the fluid mosaic or the cell membrane. They all have to have cytosol, which is also the cytoplasm, same thing. They all must have chromosomes. They have to have that genetic information that tells them what to do. And they all must contain ribosomes because that's how we build proteins. Can't make proteins without ribosomes. Okay, so we have two key types, the prokaryotes and the eukaryotes. So think about, we did the bacteria and then we had the plant and animal. They were the eukaryotes. The bacteria was the prokaryotes. So prokaryotes, domains bacteria and archaea. The DNA is in the nucleoid region because we don't have a nucleus in prokaryotes. And they're typically a lot smaller than eukaryotes are. The eukaryotes, protists, fungi, animals, plants, basically anything that doesn't fall into the prokaryote category falls into the eukaryotes. And because we have the organelles, the DNA is found in the nucleus for all of our eukaryotes. Also, they're going to have the membrane-bound organelles. So all those different organelles that we have learned about and you have learned about year after year after year, they're found in eukaryotes. So what's unique about some of these when we're looking at the different types of eukaryotes, plants have a chloroplast. That's our green, our central vacuole. So it has that big storage vacuole where we find like little vacuole vesicles smaller in the plants, in the animal cells. They have a cell wall, animal cells don't. And they have what we call a plasmodesmata as well. That's something unique to plants, but not found in animals. Animals have lysosomes, centrosomes, or centrioles is sometimes what you'll hear that referred to as, that's involved in cell mitosis. Flagella, you will often find flagella, cilia, some of those kind of things on the animal cells, and you don't typically find flagella on plant cells. So looking at the different organelles, we're going to start with the nucleus. Nucleus contains the chromosomes, which is our genetic information. It has a nuclear envelope around it, just like we have a cell membrane, we have a nuclear membrane. It's a double membrane system, and it does have pores to allow materials to pass through. You can see those little black spots. So the pores are going to regulate entry and exit of material from the cell. So they get to control what comes in and out of the nucleus. It also has a nucleolus. That's the kind of the ball or bubble inside of the nucleus. And this is where ribosomal RNA, which is the key component for ribosomes, is going to be made. So ribosomal RNA is going to combine with specific proteins and it's going to make the ribosomes. These subunits, the two pieces, will actually leave the nucleus and they will actually assemble out in the cytoplasm into the actual ribosome itself, which is where we translate messages from mRNA into that primary structure. Okay, so the ribosomes have the ribosomal RNA and protein. Those two components make them up. And they're all about synthesizing proteins. That's what ribosomes do, is they're the builders. We can find them in the cytosol just free floating. And remember, with the prokaryotes, we don't really have any other options. They're just free floating. And we can also find them bound to the endoplasmic reticulum. That's what makes it rough, ER, or to the nuclear envelope is also another place. So they kind of will attach to surfaces. And the proteins that are produced in these tend to be released from the cell. Okay. Then we have my favorite organelle, the endoplasmic reticulum. So it's membranous sacs and tubes. Basically, it's almost like a little highway or passage system. It synthesizes different membranes, and it kind of compartmentalizes the cell into sections. So, and one of the reasons we want to do that, obviously, is because we can control where different materials go if we have the different compartments. We have rough ER and smooth ER. Rough ER contains the ribosomes bound to the endoplasmic membrane, and smooth ER has no ribosomes, but it's going to synthesize lipids. It's going to help metabolize the carbohydrates, detoxify the cell, so it helps process out materials, drugs, things that the cell doesn't need to be dealing with. That's what so the smooth actually does more than the rough does, because the rough is just all about making proteins. Golgi complex. So it looks very similar to the endoplasmic reticulum, but it's more of a freestanding structure, and it's a series of membranes. And if you look, it almost looks like somebody kind of pinches it in the middle, which is what causes the curves. And we often see the little vesicle bubbles on the side because it's constantly releasing these vesicles with materials in them. I kind of think of this as the Amazon of the cell, because Amazon will take products, package them, and ship them out. And basically that's what the Golgi is doing as well. And it does have a directionality, and you'll see it tends to curve in a specific direction. So the cis face is what receives. So this flatter surface is what receives from the endoplasmic reticulum, and the trans face is the part that's actually sending these vesicles out to other membranes inside the cell or outside of the cell. So Golgi complex is all about receiving the transport vesicles with the materials, modifying, packaging, sorting, all of those things, kind of like Amazon would do. It tags them. And then once it's all tagged, it packages and ships it where it needs to go in a transport vesicle. They can exit via the membrane via exocytosis, or they can just move somewhere else in the cell if necessary. But we want to make sure that everything is properly folded, built, modified, whatever needs to happen before we package it. Lysosomes are the trash men. So they're a membranous sac with hydrolytic enzymes. So we have materials that break down parts. So hydrolyzes macromolecules in animal cells. So we've got these big macromolecules, and we need to break them into smaller pieces to be able to work with them. Lysosomes are going to do that. They can also recycle their own cells through a process we call autophagy, so they can actually engulf and break them down if they start being a problem or aging and needing to renew themselves. Peroxisomes is kind of a specific type of lysosome, so it's like a subcategory. And they're really similar, but these specific compartments will actually catalyze reactions that produce hydrogen peroxide, H2O2. And so they have a specific enzyme catalase that breaks down hydrogen peroxide into water. So it's just like a specific type of lysosome because it has a specific type of enzyme in it. Vacuoles are all about storage. So really and honestly, vacuoles are just larger vesicles. They're going to release from the endoplasmic reticulum or the Golgi, and they're selective in terms of what they're transporting. So we can have food vacuoles that form during phagocytosis. And remember, phagocytosis is like taking in something large. So we think of this as eating. And then these will get digested by the lysosomes, break it apart into the pieces we need. We can have a contractile vacuole to help maintain water levels in cells. We see that especially, there's some protists that's real common in. And then of course we find central vacuoles like in plants, that great big vacuole. And these have the inorganic ions in the water most of the time, and they help maintain a lot of the balances in the plant cell. They're really important for a thing called turgor pressure. That's what actually holds the plant upright. And so that's why sometimes if you don't water a plant for a little while, it starts to wilt, but then you'll water it and it'll pop back up. And that's because the vacuole had kind of drained and it had emptied and shriveled, but when you watered it again, it refills. Okay, mitochondria. We're going to spend a lot of time as we move through this unit and the next one dealing with mitochondria. We all know that that's kind of the powerhouse of the cell. What that's really telling us is that's where cellular respiration happens. It does have a double membrane. That's part of the evidence for that theory where endosymbiont theory, where we've brought parts into a cell and they're working together symbiotically. So the outer membrane is going to be a very smooth one. You'll see that. And the inner membrane is all squiggly foldy. So you can see the difference there. Those folds are what we call crista. And so that actually divides the mitochondria into two different internal compartments. And it also, by having all those little folds and curves, it increases the surface area where different behaviors can happen. So that inner membrane is the space between the inner and the outer. So the lighter orange space. And then the mitochondrial matrix is going to be that center dark orangey color. That's where Krebs cycle actually happens. There are enzymes that are going to catalyze that cellular respiration. And we're going to go into more specifics on cellular respiration in another unit. DNA is found in the mitochondria. That's another evidence that supports that this was actually a freestanding structure that became part of it. They have their own ribosomes. Okay. So the number of mitochondria correlates to the metabolic activity. Skeletal muscle is going to have a lot more mitochondria than say your spleen, because it has more activity. So the higher the metabolic activity, the more mitochondria they're going to have. Chloroplasts is another one with our endosymbiont theory. These are specifically designed to do photosynthesis. So they have a green pigment that's called chlorophyll. That's actually where the green coloration comes from, is that green pigment. And it also has the double membrane. Within that, we have thylakoids. And the thylakoids are each of the individual little sacs. And then we stack them together, and the stack is our grana. This is where the light reactions occur in the grana. So this is where we have to have light to make the reactions occur in there. And then we also have the stroma. That's kind of like the cytoplasm. It's the goo that all this other stuff's floating around in. And the Calvin cycle, the second half of photosynthesis, is going to occur in the stroma. This is also where we'll find the DNA for the chloroplasts and the ribosomes. These also suggest for that endosymbiont theory. Okay, so a practice free response question. Hepatitis C is a virus that attacks the liver and can cause liver disease. Liver disease has been linked to a reduction in albumin levels in patients analyzed. Albumin is an important protein in humans. It functions primarily to regulate oncotic pressure of the blood. So basically the albumin is help controlling what moves in and out of the blood vessels. Okay, on average, patients with liver disease show a 10 to 30% decrease in blood albumin levels. So first thing it asks you is identify the organelle in liver cells that is most likely being affected by hepatitis C. So we got to think about what's going to be the organelle affected based on what we know from this background. And then B, justify your reasoning. Explain why you think that's the specific organelle that's being affected. Okay. So hopefully that helps you out a little. And I'll see you in the next video. Bye.